Yeah, in today's class, we'll see the 59th question. Eukaryotic genes differ with prokaryotic genes in having. So there are some certain difference between the eukaryotic genes and prokaryotic genes. Usually we see the operons in case of the prokaryotic gene. Okay, all the genes are attached to a same promoter and regulator. Several genes are attached to a same promoter and regulator. They form just like a cluster like structure. All are attached, and three or four genes or two or three four genes, they are regulated by the same promoter and regulator gene are inhibited. So that is called as operon. For example, lactose operon or tryptophan operon. But in case of the eukaryotic genes, they act independently. Each of them are regulated by a separate promoter or regulator. And also the main difference is there is a splicing in case of the eukaryotic genes. The mRNA which is produced by the eukaryotic genes, they undergo a process called as splicing. What is splicing? Just you see this diagram. Here you see there are some genes called as exons. Some of the genes are called as introns. Exons and introns, they are all mixed, exons and introns, in case of the eukaryotic genes. So what are the exons? Exons are the active genes and introns are inactive. So during the process of maturation, what happens? All the introns are spliced out. Okay, only the exons are present in the mature mRNA. The mRNA which formed by this one, there is a process called as splicing. In the splicing, what happens? The introns will be removed. Only the protein coding parts of the mRNA only remain in this mature mRNA. So the main difference between the intron, between the eukaryotic genes and the prokaryotic genes is, in the eukaryotic genes, there are some non-coding parts called as introns. Coding parts are called as exons. Coding part means they code for the protein sequence. Amino acid formation, they are useful for the amino acid coding. So exons are the coding parts and introns are the non-coding parts. These non-coding introns will be removed. That process is called as splicing. That is the thing you should remember. That is the main difference between the prokaryotic genes and eukaryotic genes. In the prokaryotic genes, there are no introns. Okay, so that is the thing. Here they have given eukaryotic genes differ with prokaryotic genes in having exons, promoters, introns, and Tata box. The right answer is introns because both the prokaryotic genes and eukaryotic genes have exons, but only eukaryotic genes have the introns, which are the non-coding parts, non-coding sequence. Okay, that you have to remember. Next question is, which of the following statement is wrong with regard to genetic code? Now, the question is related to genetic code. We already discussed the genetic, each one is called as a codon. It contains 3 nucleotides or nitrogen bases called as triplet codon. Okay, so here they are asking for the wrong statement among the following. Which one of the following is wrong? That is the question. So we have to see here. You see the codon, genetic codon, you see here there are 64 triplet codons. Each codon consists of 3 nitrogen bases. Okay. So here you see there are 64. Out of the 64, there are 3 codons which are called as stop codons or nonsense codons or termination codons. So you see UAA, UAG, UGA. These 3 are called as stop codons or termination codons or nonsense codons because they do not code for any amino acid. But here you see there are 61. The remaining triplet codons, how many are there? 61. These 61 triplet codons code for any one of the amino acids. For example, here you see UUU. It codes for phenylalanine. This triplet codon codes for phenylalanine amino acid. And also one more triplet codon is there. UUC. It also codes same amino acid. So in the mRNA, if there is UUU, it codes for phenylalanine. In the mRNA, at the same time, if there is UUC also, it codes for the phenylalanine. It is called as degeneration of the codon because a single amino acid is coded by more than one triplet codon. That is called as degeneration of the genetic code. Okay, so you have to remember this point. Is here also leucine is the amino acid. It is coded by 4 triplet codons here. CUU codes for leucine, CUC codes for leucine, CUA codes for leucine, and CUG also codes for leucine. If in the mRNA, if any one of the 4 triplet codons given is there, this codes for leucine. This is called as degeneration of the genetic codon. That you have to remember. So next here, so what is the wrong statement here? The genetic code is overlapping. It is a triplet code. So first we sort out which one is right, which one is wrong. So as I told you earlier, genetic codon, it is a triplet code. That means it has 3 nitrogen bases or 3 nucleotides. Okay, so it is right. We have to choose the wrong one. Which one of the following is wrong statement? It is a right statement. So it is we should not choose. And genetic code is degenerate. I told you earlier. There are 61 codons which code for the amino acids, but there are more than one triplet codons which code for the same amino acid. That is called as degeneration of the genetic code. So this is also right option. This is also right statement. So we should not choose because they are asking for the wrong statement. Next, genetic code is commaless. Yeah, it is also right because there is no gap. If you see the genetic code, they continuously in the mRNA, this nitrogen bases are arranged continuously. Each 3 of them code for a amino acid. So each in the sequence, a set of 3 nitrogen bases is called as a codon. So all these codons are arranged one after one. There is no gap between the triplet codons. So it is called as commaless. Commaless means there is no gap between the triplet codons, among the triplet codons. So here here it is commaless. It is also correct statement. So we should not choose this. So among this, genetic code is overlapping. It is a wrong statement. Genetic code is overlapping is a wrong statement because there is no overlap between, among the triplet codons. All the triplet codons are they code independently for a amino acid. Okay, so this is the wrong statement. So we have to choose this as a right option because they are asking for the wrong statement in the following. So genetic code is overlapping. It is the wrong statement. And next question. What is the ratio of complementary gene interaction? So in the genetics, we have seen two types of it. Mendelian genetics and non-Mendelian genetics. Law of segregation, law of independent assortment. These are the laws proposed by the Mendel. But those which do not follow the Mendelian laws, they are referred as the non-Mendelian or non-Mendelian genetics. Here we see there are different kinds of the gene interactions. Complementary genes, duplicate genes, epistatic genes. So these are different types of the genes. They are referred as the non-Mendelian genetics. Here you see 12:3:1. This is the ratio. What is this ratio? You have to remember. Okay. In the epistasis, there are two types of the epistasis. One is dominant epistasis, another is the recessive epistasis. So this ratio 12:3:1 is related to dominant epistasis. That means one of the alleles, they mask the one allele, mask the one allele in one loci, mask the expression of the other allele in a different loci. That is called as that is a masking effect. Is called as epistasis. Dominant. So this ratio is related to dominant epistasis. Okay, and 9:3:4. This ratio you should remember. This ratio is for recessive epistasis. And there is one more ratio, 15:1. This is related to duplicate genes. And 9:7. This is related to complementary genes. So the question is related to what is the ratio of complementary gene interaction. The right answer is 9:7. So you have to go through all the ratios related to complementary genes, duplicate genes, dominant epistatic genes, and recessive epistatic genes. All this you have to remember. So complementary genes 9:7 is related to complementary genes. And next one is when homozygous red and white clad antirrhinum plants are crossed, the F1 plants produce pink flowers. You see, the parents here, there are parents, one parent has red flowers, another parent has white flowers. When these two are crossed in the F1 generation, the plants got pink flowers. So what does it mean?
Incomplete dominance.
Yes, very good. So complete dominance, wrong option. Epistasis, wrong option. Incomplete dominance. It is a mix between the two phenotypes. Okay. And one more thing you should remember, in case of the incomplete dominance, the phenotypic ratio is equal to genotypic ratio. In case of the complete dominance, you see 3:1 ratio. The phenotypic ratio is 3:1. If you see the tall variety, tall Pisum variety, tall P and short P. If these two are crossed, capital T is crossed with small t. Then what we get in the F1 generation, capital T and small t. And when it is intercrossed, self after the selfing, we get the 3:1 ratio in the phenotype. That means 3 tall and 1 short plant. But the genotypic ratio is 1:2:1. But in case of the incomplete dominance, we instead of getting 3:1 ratio, the phenotype is 1:2:1. Okay, so that genotypic ratio is also like that. So here, what we see, this is incomplete dominance. Next, in which family the stamens are obdiplostemonous? What is obdiplostemonous? You see here in the flower, how many petals are there? How many sepals are there? 5. How many petals are there? 5. But here you see the number of stamens are double. Okay, that means we see the 10 stamens. This condition is called as obdiplostemonous condition. Obdiplostemonous stamen. Okay, the number of stamens is double the number of the petals. That is called as obdiplostemonous. That is the characteristic feature of the family Rutaceae. The answer is here. You see, in which family the stamens are obdiplostemonous. That means double the number of the petals. Okay, stamens are double. That we observe in case of the Rutaceae family, that you should remember. And next one is, what type of inflorescence is present in Euphorbia? Euphorbia, there are several species. Euphorbia is the genus. Euphorbia hepta. Okay, there are different kinds of the Euphorbia species are there. In all the, and Euphorbia belongs to the family Euphorbiaceae. Euphorbia heterophylla, Euphorbia hepta. So there are so many Euphorbia species. All this genus belongs to the family Euphorbiaceae. And one more genus is there, Acalifa. Acalifa also belongs to the family Euphorbiaceae. Jatropha. Okay, Jatropha, you should remember that plant is famous for the biodiesel production, Jatropha. So all these, Euphorbia, Acalifa, Jatropha, these are the important plants which belong to the family Euphorbiaceae. And what is the inflorescence of Euphorbia? That is a question. Catkin, it is a wrong, because catkin is also present, but it is present in the Acalifa. If they ask what type of inflorescence is present in Acalifa, which also belong to Euphorbiaceae, the answer will be catkin. Okay, so and in case of the Jatropha also, the inflorescence is not catkin. Only in Acalifa we see the catkin inflorescence. In both case of the Euphorbia and the Jatropha, we find the inflorescence that is cyathium. It is a special inflorescence. Cyathium is a special inflorescence present in case of the Euphorbia genus, belong to the family Euphorbiaceae. That is the right answer. And capitulum, here one more option is given, capitulum. It is also called as head inflorescence. It is a characteristic inflorescence of the family Asteraceae or Compositae. Both are same. The old name is Compositae. The new name of the family is Asteraceae. In this family, in all the genera, we find the inflorescence that is capitulum or head inflorescence. And spike, we already discussed. Spike, it is present in the family Poaceae. Spikelet, it is also called spikelet. It is found in the family Poaceae. So you have to remember all these types of the inflorescence which are related to different genera and families. Okay, Sayatham is the answer here. Next one is, during which stage of meiosis syngetic knot is seen? So syngetic knot, it is formed between the homologous chromosomes. In the meiosis, what we see, meiosis is divided into meiosis 1 and meiosis 2. In the meiosis 1, again, there are stages, different stages. One is prophase 1, metaphase 1, anaphase 1, telophase 1. Again in the prophase 1, we see some substages. Okay, leptotin, zygotin, packtin, diplotin, all are the substages which are present in the prophase 1. So here it is very important. Prophase 1 is very important because here only the genetic recombination takes place. Crossing over, okay, crossing over or the exchange of the non-sister chromatid particles, okay, which occur in the prophase 1 of the meiosis. So it is very important. It is an important stage in the meiosis, in the entire meiosis. So you should remember all these stages. So where this syngetic knot is seen, so the right answer is leptotin. In the leptotin, we see the syngetic knot. A knot-like structure is produced that is seen in leptotin. Next, mitosis enables. What are the main features of the mitosis? There are particular differences between the mitosis and meiosis. Okay, meiosis only occurs in the reproductive cells, particularly microspore mother cells and megaspore mother cells. Meiosis occurs only in the diploid cells. Okay, meiosis occurs in the diploid cells, that too in the reproductive cells. More specifically, it occurs in the microspore mother cells or megaspore mother. Okay, that is in case of the angiosperms. But in case of the algae, meiosis occurs in the zygote. Immediately zygote. In case of the algae, the zygote immediately undergoes meiosis. So there is those life cycles are called as haplontic life cycle, because the diploid stage is limited only to the zygote. In case of the algae, those life cycles are called as haplontic life cycle. But in case of the angiosperms, meiosis occurs in the microspore or megaspore mothers, MMC. So meiosis occurs only in the diploid cells, that too in the reproductive cells. But mitosis, it occurs either in the haploid cells or in the diploids, in both the cells. So we usually refer them in the vegetative cells or in the somatic cells. We see the mitosis. So what are the main features of the mitosis here? They gave unequal distribution of the chromosomes. So it is the wrong option. In the mitosis, what happens? The chromosome number is doubled. Initially, the chromosome number is doubled and they are equally distributed to the daughter cells. So this is unequal distribution of the chromosomes is the wrong option. And next option is to maintain constant chromosome number. So that is the right option, because the mitosis, what happens? The chromosome number, I told you earlier. In the mitosis, what happens? The chromosome number is doubled in the anaphase. Okay, they are doubled, and in the metaphase, in the anaphase, what happens? The chromosome number is doubled and they are separated to the opposite poles. So they maintain, the daughter cells maintain the same chromosome number as in case of the parent cell. In both the parent cell and in the daughter cells, same chromosome number is maintained. That is the specialty of the mitosis. But in case of the meiosis, what happens? The chromosome number is, it becomes half. If there are 12 chromosomes in the parent cell, after meiosis, we find 6 chromosomes only in the daughter cell. And one more difference is, after the mitosis, from the parent cell, 2 daughter cells are produced. But in case of the meiosis, from the parent cell, after the meiosis 1 and 2, there will be 4 daughter cells. And they have the half of the number of the parent chromosome. So all these you have to see. You see, one more option is to reduce the chromosome number. So that is not the feature of the mitosis, where chromosome number is reduced to half in the meiosis.
Meiosis.
Yes, to prevent polyploidy. So these are not the right answer. What is the question is, mitosis enables to maintain the constant chromosome number in the daughter cells, in the progeny. Okay, in the daughter cells. That is the right option. Yeah. Next, what does the chromosome formula 2n minus 1 mean? We already discussed about this. If there is one chromosome less than the diploid number, 2n, it implies the diploid number. For example, in the human being, there are 46 chromosomes. That is the 2n. If anyone has 45 chromosomes, that is 2n minus 1. That is also called as monosomic. Okay. If there is 2n plus 1, trisomic. And if there is 2n minus 2, that is called nullisomic. But it is different here. What is this? It is not 2n minus 2. It is 2n minus 1 minus 1. What does it call? Monosomic? No. If it is 2n minus 1, it is called as monosomic. Disomic? No. Nullisomic? If it is 2n minus 2, it is nullisomic. And now the right answer is double monosomic. 2n minus 1 is called as, if it is only 2n minus 1, it is called as monosomic. But if it is 2n minus 1 minus 1, so it is called as double monosomic. That is the right option, right answer. Next one is, who rediscovered the Mendel's laws of inheritance in 1900? So actually, Mendel, he proposed his laws of genetics in 18th century. Okay, and more or less after 100 years later, these Mendel laws were approved. They are rediscovered, because initially Mendel laws were not approved, because at that time there was no DNA, there was no concept of the DNA. Even Mendel, he didn't know that DNA exists. DNA is a genetic material. Okay, so he proposed them as the alleles, not as the genes. He didn't know about the DNA at that time, but he was able to predict, he was able to propose the laws of inheritance. But those laws were not approved at that time. Nobody approved them that time. But later, after 100 years, in 1900 years, they were again rediscovered, and they approved that, they are approved that they are right, they are correct. Who approved them? Who rediscovered them? Hugo de Vries, Carl Correns, and Eric von Sherman. So these are the 3 scientists. They rediscovered the Mendel laws of inheritance in 1900. So you have to remember that. Next one is, what hormones regulated phototropic and geotropic movements in plants? So which hormone regulates the phototropism and geotropism in plants. We already discussed about the phototrop. The plant, while during the growth, it moves towards the growth occurs towards the light. We all know about it. Plants grow towards the light. If there is any shade, they do not grow towards the shade. They grow towards the light because they need light for the photosynthesis. And what is geotropism? The stems, they grow towards the light, but at the same time roots, they grow towards the soil, inside the soil. Geotropism. Okay, so it shows the geotropism. And which hormone is responsible for the phototropic and geotropic movements in the plant? That is the question. So gibberellins is wrong. Auxins is the right option. Auxins is the right answer. Okay, auxins. We know about the auxins. There are three important points related to auxins. One is apical dominance, and next one is phototropism. Next one is geotropism. These are very important related to auxins. Apical dominance we already discussed. Apical bud, it shows the linear growth. Plants grow straight. That is called as apical dominance. So auxins are responsible for the apical dominance, phototropism and geotropism. Next, chlorophyll A in higher plants differ from bacteriochlorophyll in having. So we already discussed. There are different types of the chlorophylls. Bacteriochlorophyll is different from the normal chlorophyll. The chlorophyll which is present in the bacteria, photosynthetic bacteria, we already discussed rhodospirillum, green sulfur bacteria, non-sulfur bacteria, purple sulfur bacteria, purple non-sulfur bacteria. These are called as photosynthetic bacteria. They have bacteriochlorophyll. And in the plants, in the angiosperms and in the algae, we find chlorophyll A. And what is the formula of chlorophyll A? C55H72 O5N4 Mg. So what is the main difference here? See, porphyrin with one pyrrole ring, no. Porphyrin with two pyrrole rings, porphyrin with three pyrrole rings, porphyrin with four pyrrole rings. Actually, here it is not properly asked the question because chlorophyll A and it has porphyrin with four pyrrole rings. Okay, and these four pyrrole rings are attached by the metal ion, that is magnesium. Okay, each pyrrole ring has one nitrogen. Since there are four pyrrole rings, it has four nitrogens. Okay, four pyrrole rings. In each pyrrole ring, there is one nitrogen, and four nitrogens are attached to a single magnesium. So chlorophyll has four pyrrole rings in the porphyrin, and bacteriochlorophyll also has four pyrrole rings in the porphyrin ring. Here the answer is given like the best or the nearest answer is porphyrin with four pyrrole rings. But here you should remember that in chlorophyll A, out of the four pyrrole rings, one is reduced. One reduced pyrrole ring is there out of four. But in case of the bacteriochlorophyll, here also there are four pyrrole rings. But out of the four, two pyrrole rings are reduced. It has two reduced eye roll rings. That you have to remember. Since they gave the answer like this, we have to choose the best or nearest answer. That is the porphyrin with the 4 pi roll. Next, integrated proteins and glycoproteins present in plasma membrane are. You know, plasma membrane. There are several theories related to that. Fluid mosaic theory, it is the most popular one. Okay, and here, as per the fluid mosaic that theory, we see there are two types. There are proteins and glycoproteins. What is the main thing here? Hydrophilic, hydrophobic, amphipathic, amphibolic. Actually, amphibolic is not at all related answer. We can exclude it immediately. It is a physics term. Amphibolic is physics term. So we have to exclude it immediately. And hydrophilic, hydrophobic. Usually proteins are hydrophobic. Lipids are hydrophobic. Usually it has a hydrophobic head and hydrophilic head and hydrophobic tail. Lipids. Lipids also called as amphipathic, and also glycoproteins also called as amphipathic because these proteins contain both the hydrophobic amino acids and hydrophilic amino acids. So here we refer them as the amphipathic. What is amphipathic? They have both the features like hydrophilic parts and hydrophobic. So we call them as the amphipathic. Amphi means both. It has both the natures, hydrophilic and hydrophobic. So that is the right answer. Next question is, which of the following is a flavo-molybdoprotein? The options here: nitrate reductase, dinitrogenase, nitrite reductase, aminotransferase. So among them, nitrate reductase, it is responsible. It is one of the enzymes important for the nitrogen fixation. So it has a